Welcome to another presentation from the CV Academics Foundation, home of the AMP Honors Program.
Hey everyone, it's Justin, one of the super mentors here with the AMP Honors Program. Today I want to discuss mRNA vaccines. With COVID-19 causing so much death and destruction, it's hard to look at the good that's come with it. The greatest thing that's come from such a terrible pandemic is the drive to find a new vaccine. Thanks to the global efforts and some amazing scientists, this feat, which usually takes several years, was accomplished in under a year. In order to understand how an mRNA vaccine differs from previous vaccines, we need to briefly go over traditional vaccines. Traditional vaccines consist of attenuated, inactivated, or pieces of a pathogen. And vaccines work by training your immune system on how to respond in the event it ever encounters the actual pathogen. The limitation of traditional vaccine lies within the amount of time it takes to produce a weakened or inactivated pathogen in the lab. Now, what if we could bypass this time-consuming step and utilize some of the machinery we already have in our cells? Enter the mRNA vaccine. mRNA, or messenger ribonucleic acid, is found within the cells of our body. mRNA is a code which tells your cells how to make a protein. Think of this like if I typed the word French fries in Word and hit print, but instead of the word printing on a piece of paper, actual food came out. In the case of mRNA, instead of French fries, a protein is encoded. Bummer. Your body is constantly using mRNA to make proteins. Usually, mRNA is made within the nucleus through a process known as transcription, and then released into the cytoplasm to be transcribed into protein. The transcription step is actually skipped in the case of mRNA vaccines because the code is already in the vaccine. Now you might be thinking, for the mRNA vaccine to protect against a SARS-CoV-2 infection, it must be making a protein found in the virus. And you'd be exactly right. Enter the spike protein. If we look here at the structure of SARS-CoV-2, you see these red things protruding from the surface of the virus. These are known as the spike protein. The spike protein is highly conserved structure throughout all human coronaviruses, not just the novel one, SARS-CoV-2. The S protein works like a lock and key. Without a key, you can't get into the locked door. SARS-CoV-2 relies on the spike protein to help it gain entry into the cell to cause infection. What makes the spike protein such a great candidate for the mRNA vaccine is the fact that it is highly conserved and in itself is harmless. Being highly conserved means even after several generations it remains the same. This again makes it a great target for vaccine production. Lastly, how did such a breakthrough in science happen so rapidly? Well, it didn't. Scientists have been studying mRNA vaccine technology for nearly 30 years. In 1990, a study was performed at UW-Madison proving it was possible to make protein by taking mRNA and injecting it into animals, utilizing its own cellular machinery. From there, researchers continued as scientists had to find ways to make a safe and effective vaccine. Now, I hope you found this video interesting. Let me know what you think in the comments below.
This has been a presentation of the CV Academics Foundation, home of the AMP Honors Program.